It gives me great pleasure to introduce one of the other organisers, Champneys from the University of Bristol. And he's going to be talking this morning on Turing instability, wave pinning, on localised patterns of cell polarity, and I'll be really three sides of the same coin.
Thank you very much. So I apologise that I wasn't here yesterday, and if John has done epsilon towards the organisation, he says, turning the mic on, then I've done epsilon to some much higher power. So I also feel like, if I'd been organised, what I meant to do is go into my shed and bring a sort of a barbecue briquette, because I feel like bringing coals to Newcastle, in that I don't really work on mathematical biology, or I only dabble, and I'm flattered to be asked to be a co-organiser of this programme, and I really would have liked to have been here, for quite a lot of these last 3 or 4, or however many months it is now. But sadly, I'm only here 3 days, and I was going to be 5, but then life intervened yesterday, and on Friday they've organised a really good workshop in engineering, which I'm also talking at. But hey, that's life. So what am I going to talk about, he says, pushing the buttons. So I'll start with just some very simple, so this is first thing in the morning, nothing complicated. I'm just looking at little toy models, mostly, motivated by various biological applications. But I did notice that the title of this week is New Mathematical Technologies or something like that. So that's my excuse for not having very much biology in what I'm talking about. And I'll start with sort of conceptual models for cell polarization, just a sort of very brief review. And then some work I finished a year or 2 ago with my colleague, Claire Grierson. I should say that all of this is the work of 2 very smart PhD students from South America, Victor Breña, who's now a faculty member in Mexico, and Nico, who's a current student halfway through from Chile. And so then we did some work on a sort of a model problem, if you like, for cellular polarity or pattern formation within the cell. And that's in plant roots. When does a root choose to grow a hair, and why does it choose to grow a hair, and where does it choose to grow a hair? And our explanation in terms of nonlinear PDE, which is the kind of theme of this whole thing, and even that is PDE on long domains, is essentially the theory of this, which kind of is interesting because I remember that about 10 years ago I was here, the last time I was here was a program called Pattern Formation in Long Domain, so probably I haven't moved on since the last time I was at the Newton Institute. And there the interesting thing is there are lots of Turing bifurcations, as you often get in reaction diffusion equations, but the critical ones are subcritical. And if you have subcritical bifurcations, that tends to lead to localization, and it leads to some interesting interplay with a spatial gradient. In plants it's always auxin, and with using some numerical results and some semi-strong asymptotic analysis, we can explain the experimental data both for the wild type and for various different mutants. Root hairs are so well established, so Claire, my collaborator, tells me, that it used to be a game to choose a mutant that would grow multiple hairs that would have various branches that would spell out the name of the journal if you actually photograph them. They're supposed to produce one hair, but there's multiple mutants for every gene. And then some results in 2D showing it the same mechanism applies. And then I'll try and bring the sort of the conceptual model at the beginning together with these results and try and wave my hands and say something a bit more general and show that actually the wave pinning idea of polarization, the Turing bifurcation idea, and this localization idea, so then the root hair you get a localized patch that then forms a single hair, can be studied in a simple canonical model that adds source and loss terms to the wave pinning model. And I'll show you with some 3-parameter bifurcation diagrams you can understand how localized patches turn into whole pattern states, and also understand how these wave pinned things turn into localized patterns. And then I'll point to future work and things I would like to learn in the 3 days that I'm here. Polarity formation, my simple level understanding is essentially that all eukaryotic cells start as homogeneous blobs and then develop some way. And the first step to developing is stopping being a homogeneous blob and being some kind of, having some polarity, have a left or rightedness. And this is how they can differentiate, form completely different shapes. This is how yeast, for example, can bud and move. And perhaps the breakdown of this can be explained, or the failure of these processes can be explained in some cancers. So there's a hypothesis, and I'm just going to assume this hypothesis. Now, I'm kind of suffering here, which I'm going to plead naivety. I'm going to plead the fifth, okay, which is naivety, in that I just arrived late last night, so I haven't heard any of the talks, and so I may say things that are just wrong and have been disproved by other people's talks, or maybe 20 years out of date in terms of experimental evidence. But my understanding is there's a growing lobby that thinks that at one level at least, the mechanism by which polarity is determined is through small G proteins controlled by gaps and gefs. And so the basic mechanism common to all these cell types across plants, unicellular organisms, and animals is some kind of interchange between these rho proteins in the cytosol and in the membrane, and there's some active step that involves activation of the bound proteins, and they diffuse much more slowly in the membrane. And so immediately you think slow diffusion of something that's bound, something in the cytosol that can move quickly. You wave your hands and produce lovely tiger stripes and any patterns you like, and produce beautiful textbooks on Turing bifurcations, perhaps. But you also see these models can do other things. So if a simple explanation of a first step, if you like, in symmetry breaking could be a Turing type instability, or it could be some kind of wave pinning, which I'll explain briefly, and or it could be some kind of localization. And I'm going to study this question with ridiculously simple models in one spatial dimension and sometimes two spatial dimensions. And you can think of perhaps the spatial dimension as representing a slice through the cell, in which case polarity formation you could think of as in terms of some front of concentration of these active G proteins, or you could think of it as a model for some region towards the edge of the cell, in which case you might expect to see some pulse-like behavior, perhaps. So there's a rich body of work, and I see Stan Marie has just walked in at the back, and he's one of the authors of a lot of this work on a so-called wave pinning explanation, which is rather different to the Turing explanation. So this is, there's a paper by Maury, Gilkine, and Edelstein-Cachette in 2008, and there's a number of other papers. And there are papers that look at interaction between the three fundamental G proteins that you get in animals and singular cellular organisms, ROS, RACs, and CDC42. I don't know who came up with these names, but it's kind of CDC42 doesn't sound quite as nice as Rho and RAC. But so perhaps we should all start calling it Fred or something, because but I know that biology doesn't really work like that. So the idea is you have a reaction diffusion equation for these membrane-bound slow, and here we've sort of homogenized the domain in some sense. We've taken this slice, so I'm thinking of a sort of a two-layer, but I'm not modeling the transverse direction. So I'm not doing something clever like Shaker does, for example, with these boundary things diffusing slowly in some boundary region, and then things diffusing quickly in the interior. I've homogenized all of that, so I've just got a single length coordinate, and the slow diffusion is of the active terms is given by this diffusion constant dA, which is very much slower than the diffusion of the inactive cytosolic G proteins, and there's some activation by these GEFs, by the gaps, sorry, and then some deactivation. But the key idea behind the wave pinning is that this is a relatively fast mechanism, and that these chemicals are, these proteins are conserved, and so you see there's no source or loss terms, and without the source or loss terms you get a fundamental instability that tends to form a front. But in some sense this model is quite degenerate, because if you looked at this model you see that it, because the kinetics is just the equal and opposite for the two equations, if you add them together and integrate and do the integration by parts business, you see that the total mass is conserved, as it ought to be in such models. And you can also see that if you do a sort of naive null-Klein analysis, you find that both null-Kleins are identical. So there's no distinguished limit of the total mass except given by the initial conditions. And what you find is essentially an invading front that stops and gets pinned at a distance that's given by the initial concentration or the initial mass of your system. And so the idea is that this wave pinning creates a sort of a left-rightedness of these activated things, and it's known that these G proteins then cause all the cellular machinery to kick in later with actin-myosin, microtubules, etc., and cell polarization actually happens. Now, some people might say, I'm studying the boring bit of cell polarization. I'm not studying the genetics, which could be thought to be the interesting bit, and I'm not studying the actual physics of the structural elements that happen when cell polarization has happened. So it's one little piece of the jigsaw, if you like, the initiation event. That's what we're trying to look at. So is this a fundamental mechanism? And here's a plug for Stan Marie and his wife, who's, his co-scientist, who he happens to be married to, who is speaking tomorrow, study lots of these questions in plants, and they'll give you some really nice stuff on pavement cells and yeast locomotion, etc. And this is contrasted with a sort of Turing bifurcation model, for example, due to Otsuji, in that this is kind of nice in that there's no wavelength involved. You don't have to sort of say how many waves can I fit in, and you know, if I have too big a domain, am I going to get too many waves? It fundamentally gives you a leftedness, rightedness, and there's some suggestion, at least in some of the models, that the fundamental instability can be subcritical, and I'll explain why I think that's important for biology. But there are questions. How does the nucleus control such a process if there's a conservation of mass? And if you think of ultimately these activated G proteins are going to be used in some process to create all these cellular structures, and also the nucleus is doing some control, then over a slightly longer time scale you might want to include small source and loss terms. And that's essentially what I'm going to get to answer to these, well, answer to this second question is what I'll get to at the end of the talk. So this is work that my colleague Claire Grierson has been doing for quite a number of years, and together with her fantastic PhD student Harry Jones, they managed to image the row proteins, which are called ROPs, which is a kind of, I know what that stands for, ROPs, R-O-P, it stands for rows of plants. So I tend to think of a nice little sort of row of plants, and if you know anything about plants, auxin does everything. So I tend to think of these little rows of plants sitting there, with auxin being like a watering can, just gently watering these rows of plants until they're activated. And that's essentially what happens. So the story of a plant hair cell. So here's a mathematician's attempt to draw a plant root. Unfortunately, as soon as I've drawn it, I need to, I'm a mathematician, so I have to obey instructions. But I'm going to disobey this, just at least for a short time, if that's okay. I don't know what it says on the back. Now, I wonder what it says on the back. It would be lovely if on the back it said, please leave. It does! Yay! Yay, great. So there's a surprise. Okay, so that's okay. I can leave this. And here's a good one for plants as well, because if I'm drawing a plant, perhaps what they're trying to tell me is that actually I should draw some leaves. So there we are, there's some leaves. A plant root, essentially, is a tip that auxin is pumped down to the tip and then pumped back out the outside. So it comes down the middle. If I draw a cross section, auxin sort of flows down the middle. This is multicellular. And on the edge of the plant root are, if I look in cross section through the root, there's a whole lot of interior cells, but the cells on the outside are, roughly speaking, divided into two types: hair cells and non-hair cells. There's slightly more non-hair cells than hair cells. And if you think of this in a frame that is moving with the tip, so it's purely tip growth, these cells move away from the tip and elongate. So they're long, thin cells, like a sort of wedge-shaped cell, which I'm drawing exceptionally badly. And they reach a critical length, and at that critical length, a patch forms. In wild type, this is Arabidopsis, of course, as everything is in plants. If you want to know what Arabidopsis is, it's a pathetic little weed. Okay, so think of this little row of pathetic little weeds that I'm watering with auxin, and then this patch forms. And this patch softens, and out of this patch all the cellular machinery kicks in, and a hair grows out to sort of somewhere out here that ends up occupying the vast majority of the surface area of this cell. The nucleus moves into the hair region because all of the work of this cell is to grow this hair for both anchorage and nutrient transport. So the question we wanted to address is, what kicks this in, and why is it always there? Well, why it's always there is you can look at a time course of what's happening to the auxin through these cells, and this is some work that Claire and her collaborators did. And they essentially showed if you look at a root hair cell and a non-root hair cell, one of the really nasties in plants is there is no technique that I'm aware of that can image auxin. You can't image it flowing. It's basically no bigger than, it's almost an amino acid. It's just very slightly different to an amino acid. But what you can do is you can image it indirectly by the in pumps and out pumps. And what they showed is that these hair cells, a very nice piece of work, the hair cells only have out pumps. And by the way, the hair cells sit in a long file on top of each other. So I have this file of cells that are getting longer and longer and longer, and the non-hair cells are like the irrigation channels because they have out pumps and in pumps. And they showed through modeling, through the work of Eric Kramer, that this essentially drip feeds. So if you look at the amount of auxin that's flowing in these non-hair cells, this is like an irrigation channel of auxin, which is drip feeding very precise amounts, it would seem, from the model of the auxin transmission into the hair cells, such that you get these traces of auxin in the hair cell. So if this is the length of a hair cell, you'll get a trace of auxin that is exponentially decaying from some peak value, and just because Victor chose this parameter, I'm going to call this peak value later K20, and this is going to be called K of x. And because auxin is being pumped and it diffuses exceptionally quickly compared to these G protein parameters, these ROPs, oh, great, compared to these ROPs, yes, compared to the ROPs, then I'm going to assume that this is instantaneous, but essentially is a parameter. So I'm going to have two parameters, which is the length of the cell and the overall concentration of auxin. Because what you find is as the cell gets longer and longer, so the overall concentration of auxin remains the same, but the profile of auxin in the cell is approximately the same. So they're going to be the two parameters we play with. And here's a very nice picture that Harry Jones took of imaging the ROPs. You can't image the auxin, but you can image the ROPs. And what you see here is, if you know what you're looking at, is some ROPs in the cell wall, and then this active patch here of the activated ROPs, and this is the point from which the root hair grows. What I haven't got a picture of, but you can sort of see here, is prior to this happening, almost all of the activated auxin that you can see is located at the apical end. So essentially we've got a process by which prior to this transition happening, ooh, I wasn't expecting that. Prior to this transition happening, there's essentially a patch of auxin at this end of the cell, that patch of the ROPs. The concentration of ROPs that you can see is at the highest concentration is at this end of the cell, and then that transitions at some point to a patch here, and this seems to occur for over many slides. There's some lovely pictures they have of this growing root hair, and then over the course of about 20 minutes it'll transition from that to that in real time. Yeah.
You said that the profile, right, that you just drew there.
This is auxin, yeah.
The auxin profile. So hardly changes with the length of the cell.
So the shape of the profile doesn't change much.
Yes.
The overall concentration.
Yeah.
So as the cell gets longer,
Yeah.
the overall concentration seems to rise. but this profile, it's basically because I've got in pumps in the neighboring channels. So I'm always sucking auxin out as I'm pumping it in from the next cell and then sucking it out from these in pumps in these irrigation channels. And there's about 10 times as much auxin here. And it's kind of interesting because auxin seems to linger in these cells to do something, but it's also needed up at the root, up at the leaves, which have now disappeared from my diagram, up at the leaves, and so there seems to flow much more quickly and in much higher concentration. Okay, so how does that happen? So we, well, in fact, Robert Payne and Claire Grierson wrote down a really simple reaction diffusion model, again, just inspired by all these row models. But now, since in this, when you look at all the sort of timescales of things happening, it seems that the unbound ROP being sort of created from the nucleus and the using up of ROP, either through unbinding or of forming the cell wall softening and root hair initiation in this growth patch where you get this patch of ROPs, seems to be important in this problem. So that allows us to include source and loss terms. So now it's not quite that this nonlinearity is minus that one. We've got some asymmetry here due to this loss term and this source term, R and B. And then we just take the simplest possible assumption. Nobody really knows exactly how this activation step, this GEF step, happens, but there's plenty of evidence to suggest that it's autocatalytic, that it's an active step, it uses energy. In the previous model there was a Hill function. The Hill function was necessary to avoid things going off to infinity. It's not necessary here because we have loss terms, so we took the simplest possible term that's consistent. And so you just get this Schnackenberg-like PDE system, but with source and loss terms and with a spatial gradient, and you see what happens. Well, Payne and Grierson simulated this model and showed that they could, well, not quite spell the names of the journals with various mutants, but changing parameters in a pseudo-intelligent way, saying, well, that would mean that I have a higher concentration of auxin, or that would mean that recycling is happening faster, and knowing what the various mutants do, they showed that this model was able to create patches like in wild type or like in mutants that generate two hairs. And interestingly, this model suggests that the second hair, the sort of one that's further away, should be more pronounced. There's a bit of cheating going on here because this is, if these look a bit like each other, these arrows show you the start and end of the cell, and these show you where the hair buds are just forming. There's some cheating, of course, because this is concentration of active ROP, which is the agent that makes the cell wall soften, and that's being compared with the bulge shape after the cell wall has softened and some growth is happening. But nevertheless, there's some suggestion, and you get these bleb-like shapes that then explode, and if you vary appropriate parameters, you can see that kind of thing. And so the model seems to have enough richness and some parameters which you can at least explain in terms of the various mutants without necessarily going down to the genetics. So if you start with a homogeneous problem, there's a non-trivial equilibrium where everything is flat, and where there's a fixed concentration of the unbound and of the bound states, and you do the usual thing with dispersion curves, etc., and you find lots of Turing bifurcations, and then you compute their criticality, you compute you do the weakly nonlinear thing, and you find that the leading ones are all subcritical. So in this particular model for the parameter values that seem to be relevant to what we're talking about, and this is just calculating, showing that we've calculated the appropriate coefficient using normal form theory. So then that got me interested. So I studied this and I thought it was something I was doing, I wouldn't say community service, I was stretching my my core expertise away from sort of dynamical systems and pattern formation, working on a genuinely applied problem. But then I thought, hang on, subcritical Turing bifurcations, that sounds a bit like what I know about through nonlinear waves. And in nonlinear waves, we're on an infinite domain, because you're talking about solitary waves typically, or in localized buckling is another area in which I've worked. But there's a kind of, sometimes it's referred to, these PDEs on infinite domains, as using spatial dynamics. So if you're on the real line, you think of space X as being time-like, and you play the game of looking for periodic waves, solitary waves, fronts, etc., by looking in phase planes and looking for periodic orbits, homoclinic orbits, heteroclinic orbits, etc. And here this is called a Hamiltonian-Hopf bifurcation of the spatial dynamics. And then we say that at the Hamiltonian-Hopf, you can understand what's happening in terms of eigenvalues of the Jacobian matrix. And I realize, I mean, I guess it's kind of obvious, but it wasn't obvious to me, that that's exactly the same condition for a Turing bifurcation. And in fact, on an infinite domain, this Hamiltonian-Hopf is the accumulation point of Turing bifurcations. It corresponds to a double root of Turing bifurcations. If you have an infinite domain, all the Turing bifurcations accumulate, and you get this accumulation point of infinitely many Turing bifurcations. And so there's a sort of, once you get onto these large domains, the sort of fundamental bifurcation in a temporal sense and in a spatial dynamic sense are exactly the same condition. And so one should expect that close to this point, the theory that occurs close to this point when you're on a finite domain should somehow go over. And it does. And one of the theories is, if you have bistability, which we do with all these nonlinearities, there's the active term is creating some multi-stability, then you should expect and the fundamental bifurcation at the accumulation point of the Turing bifurcation. So if I go back to this previous slide, these are all on a finite domain, but you can see that there's an accumulation. There's actually two families of Turing bifurcations that are accumulating on this point here. As I make the domain longer, I get more and more of them. And in this region, they're all subcritical. So what does that tell me? Well, it goes back to work I did back in the 90s. And there's a lot of people, and I see Eric's here, and I should have added his name, Eric Jochils, to this cottage industry of people looking at so-called homoclinic snaking, which is what you see in these models inside the pinning region, inside the bistability. And so that goes back to the idea of POMO pinning regions and homoclinic snaking. So here in this parameter plane of this model, this is of course without the Auxin gradient. I'm just looking at the simplest model here. This is varying the overall concentration of Auxin. There's a fundamental codimension 2 point. So this is the diffusion ratio versus this nonlinear parameter, if you like. There's a codimension 2 point where the Hamiltonian-Hopf, or the accumulation point of Turing bifurcations, changes from being supercritical to subcritical. And then you get a wedge-shaped region. In fact, it's not wedge-shaped, it's beyond all orders, as John Chapman and Gregory Kozyrev and Andrew Dean and others have shown, John King included, that this is exponentially thin in the limit as you get to this bifurcation point, but it gets wider and wider. And in here, you see infinitely many different localized solutions. You see localized patterns, if you like. There's a fundamental wavelength of a pattern. And in this model, because we've also got diffusion ratio going on, the periodic solutions have this sort of slow, fast character to them. Once you go round the loop of the subcritical, the subcritical bifurcation, there seems to be a large jump in scale, and you get these spike-like or patch-like, if you think of concentration of chemicals, clusters of patterns, and they exist on these strange bifurcation diagrams where they're all connected. Other topologies are available. And you can play the game in two-dimensional, though, because of the fundamental two-scale nature of this system, it seems that you can't find all the patterns that David Lloyd, Daniel Evertabile, and Bjorn Sandstede and others and Eric, who will be talking on Friday, have found. But that's not the real problem. So it's just showing you that I hadn't realized that a problem I'd been working on for about 3 years suddenly fitted into the framework of stuff I knew about, which was kind of a pleasant surprise. The real problem, and of course I'm giving this talk in the reverse order to we did it, the real problem has this spatial gradient. And what you find now is that this spatial gradient seems to pin the first pulse. And it also seems to mean that the first pulse occurs for a certain value of the overall Auxin concentration, whereas a 2-pulse solution, for the domain size we have, you can fit 2.5 pulses. You can also fit half pulses because we've got no flux boundary conditions. 2 or 2.5 pulse is about as big as you can get for the biologically realistic size of the domain. And what's interesting is as you vary this parameter K2, you first of all see a boundary patch, then you see something that is move towards the left-hand end, not quite 20%, it's more like 30% or 40%. But you see this slow drift into its wild-type position. So you go from this thing existing, I've now upped the concentration, this is just in steady state, sorry, I haven't done this dynamically, this is just quasi-static, and then I get a drift into this localized pulse solution. If I increase the auxin concentration, then I get a high auxin kind of wild type. And this made a new experimental prediction which they went and tested. So again, you never publish it in the order in which it's written. So they actually, Harry's result that shows that you get a boundary patch first that then jumps into wild type was motivated by what we found in this model. But as always with the biologists, they need to do more tests and everything, and they still haven't published the data. So I didn't tell you about it, and I haven't shown you any pictures, because you know what they're like. Rightly so. The other thing that's interesting, so now what I've done is something that is similar to what happens in wild type, namely that the auxin concentration grows and the length of the cell grows. So I've ramped both up, no longer quasi-statically, but dynamically and slowly. And what you see is you form a patch, but if I take the same patch solution and immediately undo this, that patch will survive all the way down to much, much lower concentrations. And the same with forming a patch and then jumping into a position that could correspond to a mutant. If I go backwards, actually it transitions into a 1.5 pulse solution. But there's hysteresis backwards and forwards. And you should expect hysteresis because fundamentally we've got subcritical bifurcations. And so you might ask, what does our bifurcation diagram look like? Well, this is a bit of a mess. But if you turn your head on its side, this looks if you turn your head on your side, this looks a bit like our snaking diagram. In fact, what we think is happening is something called slanted snaking, which is what happens, and you can easily argue essentially what happens because we've got this gradient of auxin, different parts of the domain you can think of as having quasi-static or quasi-homogeneous levels of this parameter k. So if the pulse moves into a different part of the domain, it's like I'm at a different k value in at which I might go into this pinning region. So the pinning region depends on the location in space. And there's a theory due to Jonathan Dawes of this so-called slanting snaking. So you get, and this isn't the same norm, of course, this is just, well, it is the same norm, but they don't stack up in the same way for reasons I won't go into. It's to do with the fact that I have this overall concentration of the inactive part. And so the boundary pulse exists first, then I get an overlap between where the boundary pulse exists and the wild type. That exists for a long region, but it coexists. I've only shown where solutions are PDE stable. There's lots of other branches of things that are unstable, and there's this 1.5 pulse and this 2 pulse solution. So if I were to undergo this transition here into wild type, as soon as I undergo the transition, I wouldn't transition back because I'm jumping into the middle of the existence region. And that's the fundamental benefit of having an instability due to a fold rather than a pitchfork bifurcation. And you can do all this asymptotically. You may notice because of the nature of these solutions, because we have, this parameter is going to be called delta later, which is a shame, because you can move to this so-called semi-strong interaction limit, you see that this is exactly as in the scalings of Michael Ward at UBC in Canada, his theory, and in our paper we've published a detailed asymptotic analysis that can explain all the details of this. It predicts the size of the patch. It shows that the patch will drift to some position that is dependent, the size of the pulse is dependent on its position, and that is given by the auxin gradient. So the auxin gradient is somehow controlling this position, and the reason why, going back to this original question, the reason why this is kind of only 20% of the way up the cell is precisely because we have this gradient in auxin, and that's what this theory predicts, and it gives you quite close agreement, which I hope is on the next slide, gives you close agreement between the location of this x0 and the auxin concentration. Yes?
I'm assuming Michael also got the dynamical system out of that?
Sorry?
I'm assuming Michael Ward also got the dynamical system.
Correct. We did the dynamics, not just the statics. Yeah.
What is the structure of that?
That's a good question. It's very simple. Essentially, you have a dynamical system for the slow variable, and the slow variable essentially you can show there's a single stable equilibrium point. So once you go to the appropriate slow coordinate, it's a very simple dynamical system. It's not quite as simple when you have 2 pulses and 3 pulses, and indeed you can do that. You can do it for the boundary patch, you can do it for 2 pulses, and you also get pretty good agreement. You get a 4-dimensional dynamical system rather than a 2-dimensional dynamical system if you have 2 pulses in the usual way. It's a bit like the Gorkov and Ostrovsky method, if you know about such things, but it's specific to this semi-strong interaction regime where you have this global field. The U is not localized, the V, the active part, is localized.
Just to clarify, it's not beyond all orders.
No, it's not beyond all orders. It's not beyond all orders, no. This is regular asymptotics. It's multi-scale asymptotics, essentially. You pass to a scale, but it's kind of interesting multi-scale because the U and the V are on different scales. So in the U scale, the V looks like a delta function. That's essentially what you do. And you're essentially looking at the dynamics of these delta functions moving around. And so you can produce 2-parameter bifurcation diagrams, and perhaps I won't talk to that slide. There's another long paper we've published now where I failed to correctly. I was going to put all of this in red, but clearly I put the curly brackets in the wrong place in LaTeX. But you don't need to know that. Here we consider a 2D domain, which you can think of as a projection of some surface area of this cell, remembering that actually these plant cells, to a good approximation, are essentially thin outer regions between concentric cylinders because these plant cells are mostly vacuole. There's a great big sac in the middle that keeps them at high pressure. With a plant cell, if you hit it with a pin, well, probably something a lot thinner than a pin, it would go bang and it releases its pressure. And you can do all the same game. You find transverse instabilities. So you can look, first of all, you just say, well, does the 1D theory work? Yes, but these stripe solutions, which is what you have if you just have something trivial in the y direction, they typically go unstable to transverse instabilities, and they break up into spots, which is something that you kind of would like. And you can either make these spots have a preferred position in the middle by looking at the dynamics of auxin in the transverse direction, or you can show that there is some preferred wave number that tends to lead to spots, and there's a rather lengthy calculation which I won't go into, which is in the semi-strong limit. There's a non-local eigenvalue problem that can predict the transverse instabilities. So this has now appeared, I think, in one of the SIAM journals. Okay, so let me, in the final part of the talk, the final third, I've more or less caught myself up, go back to the original problem. So what was the difference between this wave pinning model? Whoops, here's a LaTeX error. Can you see the double slash there that puts that on the second line? Yeah, it is there. It's just very faint, and unfortunately my LaTeX interpreter didn't see it. So this is the wave pinning model. The only essential difference between the wave pinning model and this auxin model, the Hill function really doesn't matter. That's it matters for the wave pinning because you need to avoid things blowing up. But once you include source and loss terms, whether you have a Hill function or a pure power nonlinearity doesn't really matter. But suppose we add, I'm very sorry about this, there's a plus alpha sitting over here and a minus epsilon theta a. So I have 2 new parameters, alpha and theta. Theta is giving me my loss and alpha is giving me my source. They were exactly the same 2 parameters that I had in the generalized Schnackenberg model for the root hair problem. And lo and behold, unsurprisingly, if I stick epsilon equals 1, epsilon is in front of these small terms, this model has exactly the same kind of structure as the Schnackenberg model we were studying for the root hair. It has a subcritical Turing bifurcation, and you can find localized patterns that are kind of semi-strong. I'm not quite in the right regime here. So here's going to be my phase diagram of this model. So let me go back here. Gamma is my nonlinear parameter. Delta is the diffusion ratio. So if you then say, well, what solutions can I get out of this model? So to begin with, I'm looking at epsilon equals 1. This is a long way from the wave pinning limit. This is nothing like the wave pinning model. So we get a region where I just get pattern states, and I get a region where I don't expect to see patterns. That's periodic solutions, if I just looked at the linear level. And between those is the Turing bifurcation. But beyond here, to the left of this green dot, is where the Turing bifurcation is subcritical. And so when I see patterns, they're fully formed and large amplitude. And I also get localized solutions existing in some region over here. And there's kind of two or more kinds of localized solutions. So I can, if I look up here, I have pattern, no pattern. But if I look down here, I see localized solutions which are like localized patterns with multiple humps. There's a point of symmetry in the middle here. Or I see things that are isolated spikes, and only numerical noise can glue them together. If I more accurate I am, the less and less I can find multi-humps. Essentially, there's a unique spike solution, and that's it when I'm down in this region. And then a little bit later, I'm going to explore and this can sort of, these spikes kind of persist all the way down to my singular limit, where the diffusion coefficient, the diffusion ratio tends to 0. But this problem, of course, has another singular limit, where this epsilon, which is multiplying these source and loss terms, tends to 0, and I go to the wave pinning model. So there's going to be some rich existence of solutions here and some rich dynamics potentially between them. So for a start, coming out of this Codemention 2 point, I get an exponentially thin wedge that only becomes numerically appreciable down here, where I have this snaking type structure, and near this point C, if I have a bifurcation diagram, I believe this is moving in the vertical direction. This dA, change of notation, is delta. I see these solution branches. I'm just plotting the active component here. I see these solution branches of these localized patterns that then eventually fill the whole domain. So I have this multi-stability between these patches of localized pattern, which look very different to what was occurring down here, where I see a spike. So the first question is, how does this homoclinic snaking localized pattern transition into a spike region? Well, perhaps I won't tell you the full story, but essentially what's happening, what is this red curve here? This red curve here is where, when I look at the far field, when I look at the possibility of what my tails are doing of some localized solution, this is where they go from oscillatory decay to pure decay. I can also see this if you're a sort of a nonlinear waves or PDE person. You can see this to where the critical point of the dispersion relation moves to some finite wavelength to 0 wavelength. And so you can understand this in terms of a change in the dispersion relation. And then if you compute the pulse type solution, you realize it doesn't mind. It doesn't care. You can use dynamical systems arguments to say, well, I've still got a two-dimensional stable and unstable manifold. I'm quite happy to exist through this transition. It doesn't bother me. But you notice there's some old theory due to Belyakov that kind of I revisited with none other than John Toland, our director of this austere, august, I should say, august institute during my postdoc. We wrote a nice paper where we talked about a plethora of homoclinic solutions, blah, blah. What you find is that as you pass through here, infinitely many multipulse solutions get destroyed. You've essentially lost the mechanism for, there's many ways of thinking of this. One way of thinking of it is when you're in the green snaking region up here, which isn't there anymore, when you're at C, you have a wavelength in the tail. And you can glue together pulses by an arbitrary number of different wavelengths apart. That wavelength then tends to infinity, and pulses get destroyed, these multipulse solutions get destroyed, essentially in some function space by disappearing out to infinity. The distance between them gets wider and wider and wider. This is complicated by the fact that I've actually got two solutions here. Because this was part of a snake, I actually have two solutions. I have a little one, which is unstable, and the big one, which is the thing you see. And what you can show, and we're currently writing up the analysis, is that these infinitely many solutions that disappear can be of the form of big guys and little guys. And so here's an image showing here of what happens to the sort of fundamental branch as you come around here. It splits into a big guy in the middle with two little guys in his tail, and the little guys disappear out to infinity. So as soon as you pass from the region where we had infinitely many localized solutions into this region where we have only pure decay, then I just have a single spike. And I have this sort of wide parameter region in here. If I go back, in here, it's not that wide, I have this region in here where I have spike type solutions, and this region out here where I have localized patterns. So how does that compare with how this diagram compare with epsilon equal to 0? Well, when epsilon equals to 0, that's a horribly singular limit, of course, because I've become suddenly this mass-conserving system. But what you can do is you can say, well, what happens to this pinning region, this region that up here corresponds to where many localized patterns exist, and down here to where I have a localized spike? What happens to this little green region as I let these source and loss terms tend to 0? Well, kind of interestingly, this little green region, which is this yellow region on this slide, change of color, it's a subset, that's all I want to show you, of the bistable region. It's the so-called pinning region inside the bistable region. That little green region is this guy here. It grows into, and this is the a codimension 2 point you can predict from bistability analysis. And this codimension 2 point and this region moves into this much larger orange region, which if you do an analysis, you can show is exactly, and there are simple analytic ways of calculating it, where the wave pinning solutions exist. So what you find is that these pulse type solutions, these spike type solutions, as epsilon, so this is taking this solution in here that happens to be in the intersection of all these wedges, and just looking at numerical continuation of that in epsilon. What was a spike, and I've used a point of symmetry in the middle, so this is my U, sorry, it was called A on an earlier slide, the active form, and this is the B, the inactive form. These become much more like two fronts. So I'm only plotting it up to a point of symmetry in the middle. And notice I get this little overshoot here, which is going to be important. When I look at this, it's not actually flat in the core. And in the V dynamics, I also get this shelf type. So the pulse turns into a shelf with this wide core. And so you might expect that this could be analyzed asymptotically. And so we can show, there are asymptotic questions. Can we explain this dip? Can we understand the width here? And how is that related to this infinite multiplicity of different widths that we could have as epsilon tends to 0, where there's complete degeneracy, and there's degeneracy between the tails here. In the limit, I'm in this pinning region, and the values of the tails are given to me by my initial conditions, rather than being, because I have this degeneracy that every point's an equilibrium point, essentially. So we did some asymptotics, or we tried to, and the first thing we did is realize that the natural coordinates to study this in are not the original U and V, which have become A and B again, but actually the sums and differences, because then we get, I don't quite know why these are double dots. I guess, what have I done here? I've thrown, something's gone wrong. These are supposed to look like PDEs. This was about 7:30 this morning that I was writing these slides, so I do apologize. And essentially what you find then is in these coordinates, you have something which has an order 1 front solution. So this becomes your wave pinning front solution, for which R becomes a parameter. And this is the natural way, in fact, to analyze the wave pinning model. And they hadn't seen it in the literature before, but it's kind of obvious when you look at it. And then the source and loss terms appear in two ways, such that R is no longer constant and such that there's this perturbation to the S equations. There's another transformation we did. What you find it's rather nasty is that the equilibrium value of this system depends on epsilon. So we did another transformation to move the equilibria R0 and S0 such that they're independent of epsilon. And so our question is, how does this pulse solution, which looks something like this when epsilon equals 1, turn into a front solution where R is exactly constant? And the answer is regular multi-scale asymptotics, but there were a few surprises and why it's hot off the press. So here's a caricature of the analysis rather than the details. The first thing is that the jump you see in the middle is indeed a wave pinning pulse, but it's not the one that corresponds to the value in the middle and the value at the end. There are two pieces, there are two shelves to this solution whose heights don't disappear as epsilon tends to 0, they just get wider and wider and wider. So the asymptotic solution is not just a front that's getting wider and wider, it's a front with an exponential shelf that gets longer and longer and longer, but the height of this shelf doesn't decay to 0, and a core, a quadratic core, again, which doesn't decay to 0, but it just gets wider and wider and wider. And you can see these in the equations. If you can see several distinguished limits. Firstly, because we're close to the equilibrium here, you move to an order root epsilon timescale as you usually do for these kinds of models, because there's, if this had been the correct slide, this would be Rxx equals epsilon, so I clearly move to a order root epsilon scale is the long scale, and an order 1 scale, I just get the front solution, and I can predict the two values of this front solution because I know what R is. R is a constant at this point. But the surprise to us is that we just assumed that because we know what this equilibrium value is, we would get the pulse solution corresponding to this equilibrium value would give us this value in the core. It turns out to be more subtle than that there is a selected front solution, but it's selected in a kind of nonlinear way by this exponential tail reaches, to begin with, an unknown point that you have to solve for. Then we get a jump, and there's an unknown width of a core, and you solve for those, and we find this asymptotic kind of solution which matches nicely with numerics, but these shelves don't disappear as epsilon tends to 0. So, concluding, because I can see John itching over there looking for his 5-minute slide. He's like the minister with his brief shuffling his papers. I've got one here that says 5 minutes, can't find it. This has been a kind of a little exercise in PDE theory on long domains using spatial dynamics, some numerics and some asymptotics, studying generic models rather than a particular biological problem. And the main conclusion, I think, is that Turing instabilities, wave pinning, and localized patterns are not, well, they're very different mechanisms, but they can all occur in the same simple models. If you look at them in the right limits, then they can all explain cell polarity. And the key is that these models of these slow reaction diffusion equations of slow membrane-bound G proteins and fast cytosolic G proteins, that mechanism is capable of producing blobs, so localized patches, localized patterns, if you want to have sort of that occurring, if you want to sort of have multiple dendrites forming, for example. It's also capable of simple left-right symmetry, depending on where you are mathematically, in which asymptotic regime you're in, essentially. And then spatial gradients, for example, of auxin, can control the location of where your blob forms or where your spatial pattern forms. And so, could nature choose? Is nature a mathematician? Well, of course not. Well, maybe she is, I don't know. But has evolution sort of chosen this mechanism because it's so rich, this G protein activation, because it's so rich and so adaptable to different formations of different kinds of patterns and polarities. One thing I would say is subcritical bifurcations are good for biology because they have this notion of hysteresis. Once you've bifurcated into some state, once this cell has decided to grow a root hair, if suddenly there was a crisis and the auxin concentration went down, it wouldn't suddenly say, oh no, I'm not growing a root hair. It makes irreversible decisions if you have subcritical bifurcations. Can explain sudden irreversible onset of root hairs and possible future work. Why I'm here to learn. Clearly these are simple models. It would be nice to think about how to make these more realistic with separate compartments for the membrane and the cytosol. It would be nice to talk to people who know the literature to find more or maybe less evidence for the ubiquity of these G protein models for cell polarization, and to apply these methods. This is going to be the second half of Nico's PhD, to apply this methodology to more realistic biology problems. Thank you.
So we have time for a few questions.
So what I find highly interesting is that we have very strong reasons to believe that the rich regime that you're describing is highly relevant, because what we do observe is that the cells are using as a bifurcation parameter auxin, as you have been showing, not only in that cell type, but in very many different cell types. But then for different cells, either it's really essential to be in the subcritical Turing regime or in the supercritical Turing regime for to get the right functional patterns. Now, it seems that these cells are using the same machinery, but are just differing in, like, you know, let's say your second parameter, which means that this codimensional 2 bifurcation, this degenerate Turing bifurcation, is actually the region around the cells are operating. So I'm wondering, so probably snaking behavior is like almost like an inevitable side effect of operating around this codimensional 2 bifurcation. Is that basically so you see it here, but so we would expect it then to be quite general, that we would observe these kind of stable, like, just like a limited number of peaks, not just one peak, but a few peaks and these transitions. Do you have more examples, or do you think there's more?
I think that was. I can remember Brooke Benjamin, who was my PhD supervisor, used to say, not so much a question, more of an observation. But that was a fantastic observation, Stan, and I know Veronica's speaking tomorrow, and I'm looking forward to interacting with you this week, both of you. Yes, it is the simple answer. I think this could be a ubiquitous, you know, in terms of bifurcation theory, you know, if you put all the ingredients there, then in some form or other, you're going to see this as the organizing center, as people used to call it way back in the 1980s, as the sort of fundamental two instabilities happening at once. There's a sort of, yes.
I noticed that you've got on your list that you're not looking to learn about matched asymptotics, but I want to throw in an observation. The slide at the end,
Yes.
bring that back. So it's a question, but I think it's also a prediction. So isn't it the case that the role of the boundary layer is to provide not a Dirichlet boundary condition, but a second derivative boundary condition, which then gives this interpolation between the expected value at the midpoint and this value around which the transition occurs?
I don't know, but I'd love to talk to you about it. So yes, that you can add that to my list. There wasn't room on the slide to say learn some more asymptotics.
Maybe one more question.
From mathematical point of view, you could have a reaction diffusion system of activated distributed type. Okay, you could have two equations without source or loss term or inhomogeneous terms, actually. Okay, you could have stable localized solutions such as house from could be standing one or triune solutions. And would this be?
I think what's important here is that it's Schnackenberg-like not Gray Scott-like. So I'm aware that there are lots of different families of reaction diffusion equations, and depending on what your question is, you can find all kinds of different dynamics. I'm aware of some of that literature, but we've tried to be a little bit motivated by the biology of using the ones that are suggested by the biology. But I'm happy to talk.
Okay, I think we move on. Thanks, Alan, again.